Hello and welcome to you all to our first event of 2022. For those of you who don't know me, my name is Gavin Merrifield and I will be hosting you for this event tonight. As you will have already have noticed, we are trying a few new things out tonight. The most obvious of these is that we are viewing tonight's event as a live broadcast on both YouTube and Facebook rather than our usual Zoom. This should make things simpler in terms of finding our events in future and also improving the presentation that we bring to you. Hopefully all will go well tonight, but we may experience a few technical gremlins along the way. If we do, stick with us and we'll get things sorted as soon as possible. As ever, tonight's event has been organised by the Manchester Science and Philosophy Group, who are the local group of Christians in Science here in Manchester. Our events are from a Christian perspective, but anyone from any background with an interest in science, religion and faith is very welcome. The CIS is an organisation that brings together and supports those interested in the positive interactions between science, the church and Christian faith. You can go to our website at www.cis.org to find out more about us. And if you would like to support the wider work of CIS, please consider whether you would like to join us as a member or as a friend of CIS, or even think about affiliation for your church. Details for all of these can be found on the website. When it comes to the discussion time after our main presentation tonight, please enter your short comments and insightful questions into the appropriate comments boxes on either YouTube or Facebook. And we will try our best to get those answered for you with our speaker. Just before I introduce him, a reminder that to stay up to date on our future events, you can follow our Eventbrite page, or you can ask to be added to our mailing address list. If I can get this to work, the address should be appearing now. So send us an email there to be added to our mailing list. So our upcoming events, we've got two more planned after tonight, all beginning at 7:30. On February the 25th, we have Steph Bevan, who is both a Christians in Science Development Officer and a Public Engagement Manager with the Institute of Physics. And she'll be speaking to us under the title, Planets, Picaloos and People, Living the Way We Are Made. In her talk, Steph wants to explore some of the things that have inspired her awe and wonder in the world, and why this has become important to her own faith. Be prepared to take part and be interactive in this event. Steph very much likes plenty of participation and is looking forward to hearing your own stories about awe and wonder and the impact it has had on your own lives too. Then on the 31st of March, we are going to be joined by Joshua Smith, who will be joining us over the interweb from the USA to talk about his new and very exciting book, Robot Theology. So really hope you can join us for both of those events. This evening, though, we are giving a warm welcome to Dr. Neville Cobb. Now, Neville, as you can see here he is. He's a good friend of mine from our shared days back at the University of Edinburgh, where we worked in opposite buildings, but both doing research on invertebrates. In my case, hairy giant spiders. In Neville's case, the actually much more useful Drosophila fly. Neville and I also used to run the Edinburgh local group of Christians in Science together for many years, and that was a great opportunity to see his deep and careful interest in all things science and faith, from the Hebrew language to genetic chimeras. He is quite diverse. After Edinburgh, he spent some time in Liverpool doing further research before changing professional tack when he'd begun training for Christian ministry over in Northern Ireland, currently with the Church of Ireland, and that's where he joins us from tonight. A word of warning, though, I have recently seen this man give a very stern Burns Night address, keeping at bay a pretty fearsome-looking haggis. I'll just say, well, you've been warned. Due to some possible connection problems at Neville's end, he's provided us with a video of his presentation, and then he will join us again after that video, where he will answer questions live for us. The main presentation is about an hour, and then we'll move into a discussion time of about 15 to 20 minutes after. So settle in with your cup of tea and your biscuits. As ever, if you have questions or comments, feel free to post them as the evening goes on, rather than just storing them up towards the end. and possibly forgetting them. So without further ado, Neville and I will vanish and we'll play the video for you. So we'll rejoin in about an hour.
Chance and Necessity. I've taken the title for this presentation from an influential book published in 1970 by the French molecular biologist Jacques Monod. Jacques Monod is probably best known for his work on the lac operon, together with his collaborator François Jacob, for which they both received the Nobel Prize. They laid the foundations for much of our current understanding of the regulation of gene expression and postulated for the first time the existence of messenger RNA. In his book Le hasard et la nécessité, translated into English as Chance and Necessity, Jacques Monod in turn drew inspiration from the Greek philosopher Democritus, who described how everything existing in the universe is the fruit of chance and necessity. At the conclusion of his book, Jacques Monod wrote the following: L'ancienne alliance est rompue. L'homme sait enfin qu'il est seul dans l'immensité indifférente de l'univers, d'où il émergeait par hasard, non plus que son destin, son devoir n'est écrit nulle part. Translated into English, the former covenant is broken. Man at last knows that he is alone in the unfeeling immensity of the universe, out of which he has emerged purely by chance. Neither his destiny nor his duty have been written down. Now, it may not surprise you to hear that Jacques Monod was an atheist. Similar views were later expressed by the American paleontologist Stephen Jay Gould, based on a number of computer simulations which he performed with collaborators in the 1970s. Stephen Jay Gould came to the conclusion that if you could replay the tape of life, obviously this was in a pre-digital age, but if you could replay the tape of life, you wouldn't see the same outcomes occurring, the same species emerging, the same structures evident. But rather than describing Stephen Jay Gould's views at length or reading out from his books, I'm going to let you hear him speaking in his own words in a collection of representative video clips.
We want to know why we're here. To a large extent, it is a grand-scale accident that we're here. Evolution has oddly contingent pathways. It would never run the same way twice. If you could go back 500 million years and run the tape of life again, you wouldn't get human beings, and you probably wouldn't get anything conscious. So the accident is the 60 trillion contingent events that eventually led to the emergence of Homo sapiens. It's that no species now alive is predictable, and anyone that's here is here by the merest good fortune of tens of thousands of antecedent events which went one way and not the other. If the dinosaurs, as we were saying, hadn't died off, we wouldn't be here. Mm-hmm. If the fish didn't have the strong central axis, they never would have had terrestrial vertebrates. If a different set of creatures had survived this decimation pattern of the Burgess Shale, we wouldn't be here. If the eukaryotic cell had never evolved through this odd mechanism of symbiosis, there probably never would have been more than algal mats. Any lineage that's here, including our own, is here more by the luck of this grand cosmic draw than by any predictable necessity.
So that's the view of Stephen Jay Gould, which, as I'm sure you can appreciate, is rather similar to that of Jacques Monod in arguing that evolution is essentially a chance process, that the origin of life is essentially more due to chance than to anything else, to happenstance. Has anybody heard of this person? Do you think that he might express the same view? Well, some of you might be surprised. Let's hear what he has to say for himself.
I strongly deny that evolution is random selection. Evolution is non-random selection. There have been people in my field who have tried to say that evolution is a kind of random walk, and I think that's clearly wrong, and I'm going to try to persuade you of that. Random genetic variation and non-random survival and a non-random reproduction, which is why, as the generations go by, animals get better at doing what they do.
Let us hear the conclusion of the whole matter. The essence of life is statistical improbability on a colossal scale. Whatever is the explanation for life, therefore, it cannot be chance. The true explanation for the existence of life must embody the very antithesis of chance. The antithesis of chance is non-random survival, properly understood.
So there you've heard what I trust is a representative sample of Richard Dawkins' views over an extended number of years, concluding with him reading from his own book, first published in 1986, The Blind Watchmaker, in which he articulates how evolution by natural selection is not a purely random or chance process, differing in this respect from either Jacques Monod or Stephen Jay Gould. Now, the next person we're going to hear from is not an atheist, but rather a devout Christian, a member of the Church of England, Simon Conway Morris, who wrote this book, Life's Solution, in 2003. But rather than giving you a second-hand account of Simon Conway Morris's understanding, let's hear from him in his own words as well.
Well, going back to the ideas that neo-Darwinian explanations say all things are possible, humans and everything else are evolutionary accidents, well, that may be true, but on the other hand, when you find a recurrence, when you keep on returning to the same solution again and again and again, it does suggest that there is, at the very least, a deeper pattern there. We are dealing with a rather precise set of arrangements, and the number of alternatives, as I think evolutionary convergence shows, is a hint that there are deeper organisational principles at work. The more we look at biology, and especially evolution, I'm more and more persuaded actually that there is a deep structure to it.
So there you have a flavour of the contrasting perspectives. Simon Conway Morris as a Christian, Richard Dawkins as an atheist. Both have an understanding of evolution by natural selection as a process that has a pattern that can be recognised. Not something that is purely down to chance, not something that is purely random, in contrast to the understanding of either Jacques Monod or Stephen Jay Gould. You'll have noticed how Simon Conway Morris makes reference to evolutionary convergence. This isn't a new concept. In fact, Charles Darwin first mentioned this, writing in chapter 13 of his book on the origin of species by means of natural selection. He described how animals belonging to two most distinct lines of descent may readily become adapted to similar conditions and thus assume a close external resemblance. We can see examples of this even in species that we can find in the British Isles.
Two species that you will find within the British Isles are Glombulus marginata on the top, which is one of the myriapods. It's a millipede. On the bottom, you have a crustacean, a type of woodlouse, Armadillidium vulgare. Both of them can roll up like an armadillo. Very, very similar in appearance, but in terms of their relationships to each other, they probably diverged by about at least 550 million years, going back to the Cambrian. So they're not closely related, but they've very clearly got a very similar external body plan, very similar strategy for survival. Similarly, you can see this in plants, very similar body plans being shown by a cactus on the left-hand side, Astrophytum asterius, and then a euphorbia obesa, on the right-hand side. Very, very similar appearance, but not closely related to each other at all. We can see this also in the fusiform or a spindle-shaped body plan with a similar arrangement of caudal fins, pectoral fins, pelvic fins where available, and the anal fin. On the top, you can see a shark, one of the cartilaginous fishes, the chondrichthyes, a bony fish, shown here by a marlin. Not closely related to each other again. These are separated by millions of years of evolution, hundreds of millions of years. Then an ichthyosaur, reconstructed down here, a reptile dating from the Mesozoic period, and the dolphin, Tursiops truncatus, the bottlenose dolphin. Not closely related to each other, we've got a mammal, a reptile, and two types of fish. Very similar body plans, unrelated organisms. All examples of convergent evolution. I'll whip through a few more examples of these. How many crocodiles do you think you see here? 4, 3, 2, 1. The only crocodile you can see here at the top, or the only member of the crocodilomorphs, the crocodile family, is the top one. This is a gharial, or gavial. This one here is another reptile. This one dates from the Cretaceous period into the Eocene. This is Champsosaurus, one of the Charistidaira. Not closely related at all. This one at the bottom here is a phytosaur, which you can distinguish by having its nostrils on the top of the head instead of at the rostrum at the tip of the snout. And this one is actually Ambulocetus, a prehistoric whale, which nevertheless adopted a crocodile-like lifestyle, living in rivers. Not closely related to each other, but all very, very similar body plans. You can see this also in the evolution of flight, birds, bats, pterosaurs, and arguably flying fish as well. Flying fish could be argued to be gliders rather than true flyers, but they're capable of powered flight insofar as they're able to launch themselves out of the water by swimming at great speed, and they can glide for a far further stretch than any other animal. Each of them has an aerofoil formed by either the forelimb or the pectoral fins in the case of the flying fish, Exocetus. All of them showing clear examples of convergent evolution, wings being formed in slightly different ways, but nevertheless all forming an aerofoil by which they achieve flight. If we take true gliders, organisms which are undisputably gliders, various gliding mammals, at the top here we have a colugo. Here we've got a flying squirrel, and this is a sugar glider or a flying phalanger. This is a rodent, the flying squirrel. This is a marsupial, the flying phalanger. And the colugo is arguably more closely related to both rodents and primates. It's not really either. Again, among burrowing animals, we have the European mole up here and the golden mole up here. The European mole is an insectivore. The golden mole is an afrotherian. It's more closely related to elephants than it is to us. The mole is more closely related to us. And this one at the bottom, which looks remarkably similar to the golden mole, is the Australian marsupial mole. Not closely related to each other, all very similar body plans. Similarly with spiny animals, the familiar hedgehog, which is an insectivore, the porcupine, which is a rodent. Both of them belong to the group of mammals known as Laurasiatheres. The tenrec, which looks very like the hedgehog found on Madagascar, the greater hedgehog tenrec. This is actually one of the afrotherians, again more closely related to elephants, hyraxes. And this one, the echidna, is actually a monotreme, an egg-leg mammal. Not closely related to each other, very similar pattern of defending themselves by spines. Even more remarkable is when we look at the thylacine at the bottom here, which is now extinct, the Tasmanian wolf or Tasmanian tiger, and the timber wolf, European wolf or North American timber wolf. If you look at their skulls, they're almost indistinguishable from each other. Organisms, again, that are not closely related to each other. So there's clearly a pattern, there's clearly some constraints on the evolutionary process, that you get these very, very similar structures forming from unrelated organisms. The only way you can really distinguish these skulls is you look at slight differences in the articulation of the jaws, extra holes in the inside of the palate, and if you count the number of incisors, you'll notice they're different between the thylacine and the timber wolf. You can find this as well in the raptorial way of preying, as you can see in a praying mantis, or the mantis fly, which is actually more closely related to lacewings, and the mantis shrimp. Similar ways of obtaining food, unrelated organisms. This one isn't an insect at all, of course, it's a crustacean, more closely related to crabs and lobsters. And another example, extinct now, is the saber-toothed killing form, the saber-toothed means of killing. So the example you can see here is Smilodon fatalis, the most famous of the saber-toothed cats. Not a closely related organism is Barbarophilus, which is far more distantly related. It's not a true cat. Barbarophilus, again, has these extremely extended canines, adaptations for stabbing, stronger neck muscles, weaker jaw muscles, but a very powerful stabbing action. Similarly, the nimravids, which are even more distantly related among feliform predators, such as this one, Hoplophonius, and even more distantly related again, not even a carnivore in the sense of the order Carnivora, to which bears, dogs, cats, etc. all belong. This is a creodont. Creodonts didn't have any chewing molars. They all had shearing teeth in their jaws. This is a creodont called Macharoides, which again has these extremely elongated upper canines for stabbing. Even more distantly related, we have a cousin of the marsupials, the Sparacidonts, Thylacosmilus, which you can see again these stabbing canines, highly elongated stabbing canines.
And even more distantly related among early mammal-like reptiles, the therapsids such as the Gorgonopsids, this one's Inostransavia, and you can see these very enlarged stabbing canines. Very, very clear convergence among distantly related animals, so clearly there is some pattern involved.
Probably one of the best examples of convergent evolution, though, is looking at the camera eye. You can see clearly here the similarities in the structure of the camera eye in humans, like ourselves, and a cephalopod such as the common octopus, Octopus vulgaris. The only differences, really structurally, are the way the optic nerves exit from the eye, and the way that the lens is focused is slightly different as well. It moves forward and back instead of being stretched to change its shape in the case of the cephalopods. The camera eye can also be seen in other organisms, such as this alciopid worm, the polychaete worm from the phylum Annelida, more distantly related as well, but you can clearly see its camera eye structure, these very large camera eyes. And more remarkably, the box jellyfish, far more distantly related. It has structures called rhopalia, which you might be able to see around the corners here, 4 of these. And these rhopalia have 2 camera eyes within them, as you can see here. Now, quite why an organism that doesn't have a central nervous system needs to have such visual acuity is a good question, which still remains unanswered. But nevertheless, it clearly has a survival value, otherwise it wouldn't be retaining them. So camera eyes among very, very distantly related organisms are clearly conserved as a structure. You can also look at convergent evolution at the molecular level, looking at patterns of evolution in DNA sequences. And in this case, it's looking at a number of organisms which have echolocation, bats and dolphins, and looking at genes which, in terms of their phylogeny, their evolutionary patterns, seem to be more closely related to other echolocating species, be they other echolocating bats or bats and dolphins together, rather than the true organismal phylogeny, the pattern of evolution of all of the genes in the organism by and large. If we're to look at molecular evolution at the level of proteins rather than DNA, convergent evolution can be a little bit harder to spot sometimes because protein sequences tend to be far more conserved at the structural level than at the sequence level. Nevertheless, this is some work I was doing on the conservation and the evolution of kinases. I was looking at the radiation of kinases, enzymes that transfer phosphate groups
to other proteins in particular, sometimes to other molecules. This is an evolutionary tree of all the kinases in humans. If we focus in particular on the branches shown in blue, these represent kinases that transfer a phosphate group to either a serine or a threonine amino acid. The top asterisk refers to the Wee1 protein kinase, which in humans and flies transfers a phosphate group to a tyrosine residue, even though it's more closely related to other serine and threonine kinases in the same organisms. Similarly, the pseudopodium enriched atypical kinase 1 clusters among the serine-threonine kinases, but has intrinsic tyrosine kinase activity. Similarly, lemur tyrosine kinase 2 only exhibits serine-threonine kinase activity, despite being most closely related to tyrosine kinases.
So something that was more closely related to the serine and threonine kinases has convergently evolved the ability to transfer a phosphate group to a tyrosine. And conversely, proteins that are more closely related to tyrosine kinases evolve the ability to transfer the phosphate to serine and threonine. You can see convergent evolution in proteases as well. So on the left-hand side, we have a number of serine proteases that perform proteolytic cleavage at a serine amino acid or at a cysteine, in the case of the ones on the right-hand side. Subtilisin from the bacterial species Bacillus subtilis, prolyl oligopeptidase. These are both serine proteases, but you have a very similar catalytic triad in these unrelated proteins, which are structurally not that similar to each other as well, but a very similar catalytic triad of aspartate, histidine, and either cysteine or serine conserved between the serine proteases and the cysteine proteases. In the case of plants, most of the examples I've discussed so far have been animals. In the case of plants, there was a paper looking at convergent evolution in carnivorous plants, pitcher plants, which are not closely related to each other. So you have the Australian pitcher plants being shown at the top here. Then you have Nepenthes, North American pitcher plants, and Sarracenia at the bottom here as well. Some pitcher plants from Asia. Nevertheless, although these plants are not closely related to each other, there are actually some carnivorous plants more closely related to them than they are to each other. They've all independently evolved sometimes very similar enzymes, proteolytic enzymes and chitinases, to digest the exoskeleton of insects made up of chitin.
However, the most remarkable example of convergent evolution in plants is probably the C4 photosynthetic pathway. Now, most of you have probably heard about C3 photosynthesis before, even if you haven't heard it called that name. C3 photosynthesis is what most people learn about in school. During C3 photosynthesis, an enzyme that we call rubisco for short combines carbon dioxide with ribulose 1,5-bisphosphate to generate 3-phosphoglycerate, which is a 3-carbon molecule, hence C3 photosynthesis. However, in the C4 photosynthetic pathway, by contrast, we have an enzyme called phosphoenolpyruvate carboxylase, which combines carbon dioxide with phosphoenolpyruvate to give you a 4-carbon molecule called oxaloacetate. Now, in plants that have C4 photosynthesis, there is a rather unusual anatomy in the leaf of the plant, which we can see here in this example from maize, or Zea maize. Here we can see that the leaf veins are surrounded by cells that are particularly rich in chloroplasts, which are called bundle sheath cells. The bundle sheath cells, in turn, are surrounded by mesophyll cells. Now, the mesophyll cells are the cells where carbon dioxide is fixed by phosphoenolpyruvate carboxylase. The oxaloacetate, which is produced as a consequence, is then changed into malate. The malate is shunted into the bundle sheath cells, and it's in the bundle sheath cells where the light-dependent reactions occur. So the light-independent reactions of carbon dioxide fixation are separated from the light-dependent reactions, which generate glucose. C4 photosynthesis tends to be far more efficient under conditions where you have high light intensity, but also high temperatures and a limited water supply. So plants tend to close their stomata to prevent loss of water through transpiration, but also tend to have a limited supply of carbon dioxide as a consequence. C4 photosynthesis is therefore more efficient in preventing the unwanted reactions of photorespiration, where the supply of carbon dioxide is limited under conditions of high temperature and low supply of water.
This pathway of C4 photosynthesis, which involves a major anatomical rearrangement as well as changes in gene expression, this has evolved, it's been estimated up to about 60 or more independent times in different lineages of plants. Very, very clear, strong example of convergence occurring.
So, as we're able to see from all these many examples of evolutionary convergence, there is clearly some kind of pattern to the evolutionary process. There is clearly some kind of constraint. Now, one way this can be examined experimentally has been conducted at Michigan State University in the laboratory of Richard Lenski and his co-workers, something known as the long-term evolution experiment using the bacterium Escherichia coli. They started work back in 1988 on a number of populations
of E. coli, where they were growing them in a very, very simple minimal media containing just a few salts, the glucose solution, and they also had some citrate present to try and collate the iron to allow the bacteria to take that up as well. So they originally had these divided into 12 different populations, some of which could utilize arabinose, some of which didn't, labeled as ARA- and ARA+, 1 to 6, so 12 in total. And looking at earlier generations of these, so this is looking at about 10,000 generations, one of the lovely things about this setup is that they could freeze evolution in time. They could freeze down the bacteria as glycerol stocks, put them in a -80 freezer, and then resurrect them at a later stage and start competing the descendants with their ancestors and measuring their relative fitness. What they could see, the early generation here, the relative fitness is obviously 1, it's unity because it's comparing the same thing with itself. But if they look at all of these 12 independent populations of bacteria, they're all converging on a similar increase in relative fitness compared to their ancestor. Similarly, the size of the bacteria seems to be increasing. They seem to see a convergence and an increase in cell volume. So experimentally here, as well as just forensically trying to go back in time, experimentally here we can see a clear case of convergence.
And also, if you're to look at the patterns of gene expression, this is later work that they were able to do when microarrays became available to look at levels of gene expression. If they compare two different ancestral lines with each other, here they're measuring the sort of divergence in patterns of gene expression, 1 minus the correlation coefficient is what this number is. You're effectively seeing very, very little difference between two different ancestral lines. If you compare an ancestral line to its descendant, you're seeing far more difference, both B and C. But if you compare two descendants to each other from two different independently evolving lines,
they are more similar to each other than they are to their ancestors individually. So convergence is clearly reflected in the patterns of gene expression as well.
Richard Lenski and his group also then looked at the extent to which the genome is evolving in just one of these particular lines.
Taking one particular population in isolation, looking at it through successive generations, moving outwards here, they could see inversions of the chromosome occurring, but lots and lots of different point mutations occurring, which are shown in purple. The interesting thing was all 26 point mutations that they examined produced substitutions in amino acids. In other words, this is an example of positive selection, where they had no synonymous substitutions, those which didn't change the coding sequence. All of them were non-synonymous. So clearly very, very strong positive selection. This isn't neutral, this isn't negative selection. They could also compare, however, the frequency of parallel mutations occurring in 11 of the others. So where they observed those mutations in that particular strain, they could see, well, how many of these mutations are also occurring in the 11 other independent lines. 3 of them were found to be completely conserved. Another 9 were found in some of the other populations, and only 2 didn't appear in any of them. So completely independent bacterial lines, all evolving convergently. One interesting thing emerged later on in the study, though. They had one population that was able to utilize citrate. So the citrate was originally provided, the citric acid in the culture, only so they could take up iron. But one population independently evolved the ability to utilize citrate, and that one became quite cloudy in the culture because it had an extra carbon source, not just the glucose that was provided. Now, with this citrate-utilizing population, they only saw convergence in later stages of the study, so over subsequent generations. If they tried to go back to the earlier populations that they'd frozen down, up to about 15,000 or so, they're not seeing any independent evolution of citrate utilization, but the later ones do. So here we're seeing 2 things. We're seeing convergence, yes, but only later on, but also a clear element of contingency, which is one of the things that Stephen Jay Gould emphasized, the contingency of evolution. And they described how there might be different mutations occurring here in this particular paper, some which are potentiating mutations, which make citrate utilization possible, then an actualizing mutational event, which makes the phenotype manifest, that they're now able to utilize citrate, and then maybe some subsequent refinement mutations, which make citrate utilization even more effective. So among those citrate-utilizing bacteria, they could see various rearrangements where the gene that is transporting citrate, the gene that encodes the protein responsible for that, the citrate succinate antiporter, in other words, citrate in, succinate out, that is normally not expressed when bacteria are growing aerobically in the presence of oxygen. But a rearrangement had occurred in all of these bacteria such that they now had an aerobic promoter in front of this gene. So basically they could use citrate in the presence of an oxygen in the environment. And very similar arrangements were seen in all of these independent lines of bacteria that were able to utilize citrate. Somehow a promoter element that would allow citrate utilization was in front of this CYP gene. So, strong evidence of convergence, but also some contingency.
So we can see that the process of evolution is clearly constrained. It is constrained both by history, in terms of contingency, and by environmental factors, as reflected in the multiple examples of convergent evolution. So if evolution is not completely random, but is rather constrained, then one should be able to make some predictions about the course of evolution. And one of the earliest predictions was made both by Charles Darwin and by Alfred Russel Wallace concerning this plant, a particular species of orchid called Angrecum sesquipedale. This orchid has extraordinarily long spurs of about 1.5 feet long, hence the name sesquipedale, at the very bottom of which is nectar. Now, neither Charles Darwin nor Alfred Russel Wallace were aware of any particular species of insect that had a proboscis long enough to reach down to the very bottom of these spurs. But based on an understanding of coevolution between plants and their pollinators, they expected that such an insect must exist. In a paper that Alfred Russel Wallace published, defending Charles Darwin's theory of evolution by natural selection, he included this illustration of a hypothetical African hawk moth, which was effectively like a wanted notice for the pollinator of Angrecum sesquipedale. However, it wasn't until long after both Charles Darwin and Alfred Russel Wallace were dead that the moth was finally discovered. This is Xanthopan morgani praedicta, so called because it was predicted by both Darwin and Wallace, based on an understanding of coevolution. An understanding of coevolution can also allow us to make other predictions.
Molecular coevolution also allows us to make predictions. This is a paper I published a number of years ago, looking at the evolution of SMC proteins, which are required for compacting DNA as chromosomes divide. And they're in pairs with each other, such as SMC1 and SMC3 are found together, SMC2 and 4 are in a complex together, and they co-evolve with each other. And by looking at the patterns of coevolution, it allowed me to distinguish between two different possible models. So whether or not these two proteins in any individual complex might combine intramolecularly like this or intermolecularly like this, and looking at patterns of molecular coevolution along the branches of that tree I've just shown you, you could clearly distinguish between the models in a way that was consistent with the experimental data from protein crystal structures.
So as you can see, the process of evolution by natural selection is not completely random. On the contrary, we can discern various patterns which allow us to make reasonable predictions. One of these predictions is named after this chap. This is the Belgian paleontologist Louis Dollo, after whom Dollo's law, or as some people refer to it, Dollo's principle, is named. And Dollo's principle, or Dollo's law, concerns whether or not evolution is reversible. And how might this relate to the patterns that we see in evolutionary convergence? Well, Richard Dawkins, in his book The Blind Watchmaker, defines Dollo's law in this way. He says it is really just a statement about the statistical improbability of following exactly the same evolutionary trajectory twice. And as such, Dollo's law is so obvious that nobody needs to test it experimentally. Nevertheless, some people have tested Dollo's law experimentally.
This is looking at glucocorticoid receptors and trying to estimate using maximum likelihood analysis. So basically going back and trying to predict what is the amino acid sequence of the ancestor. So you have all these glucocorticoid receptors, which are sensitive only to cortisol. These ones are sensitive to other hormones, cortisol and mineralocorticoids. And they've worked out here a potential ancestral sequence at different points, the ancestor of all the glucocorticoid receptors that are only sensitive to cortisol, and the ancestor of those that are sensitive not just to cortisol, but also to aldosterone and other mineralocorticoids. Not only were they able to try and estimate what the amino acid sequence of these proteins were, they made the proteins and also produced crystal structures of them. And then we're able to identify particular amino acid substitutions that change the position of this helix 7, for example, this particular amino acid, a leucine converted to a glutamine, and other mutations that might stabilize the structure. So these are the responses of these different receptors to different hormones. The ancestor of all the glucocorticoid receptors, ANCGR1, where you can see responding to, in purple, these to cortisol and also to the mineralocorticoids, whereas this one, the ancestor of all the ones sensitive to cortisol only, is only sensitive to cortisol in purple. If they tried putting in the amino acid substitutions that seem to be changing the structure and stabilizing the structure between these two, introduce these into this background, they then lost the sensitivity to mineralocorticoids such as aldosterone. Still had some to another hormone, deoxycorticosterone. So when they, on the other hand, tried to do reverse substitutions into this later ancestor, they seem to lose sensitivity to any hormones. So this seemed to be suggesting, in agreement with Dollo's law, that evolution can't go backwards. But then they also started looking for some maybe additional substitutions, looking for some extra amino acids that seemed to be completely conserved among all the glucocorticoid receptors, and some that were only conserved among those sensitive to cortisol, and identified a number of about 5 other additional amino acids. And when they mutated these ones in addition, they then could restore the ability to respond to mineralocorticoids in the later ancestor. So, evolution is reversible to some extent.
However, Louis Dollo himself described the principle named after him in rather different terms to those used by Richard Dawkins. According to Louis Dollo, an organism is unable to return even partially to a previous stage already realized in the ranks of its ancestors. So rather than being a statement about the statistical improbability of following exactly the same evolutionary path twice, Dollo states that an organism is unable to return even partially. Now that, we know, is not the case.
Take the example of phasmids, stick insects. Most of them are unwinged. They're wingless insects. Some of them have small, stubby wings. Some do have full wings and are capable of flight. If you look at a phylogeny, an evolutionary tree of the phasmids, the vast majority are wingless, but compared to other winged insects that they're closely related to, it's fairly clear that the most parsimonious, the simplest explanation for the pattern you see in this tree is that the winged state has been regained later on, multiple times. So somehow, phasmids were predominantly wingless, evolved from winged insect ancestors, but some of them have regained the ability to produce wings. Clear contradiction of Dollo's law. An even more striking example concerns a few frogs of the genus gastrothica, which have teeth in their lower jaw, mandibular teeth. Most frogs don't have mandibular teeth in the adult stage compared to other amphibians, but these ones down here do. So there are conditions where, if it is favorable for an organism, if there is a selective advantage, evolution can sometimes go backwards. Which leads me on from this slightly humorous slide to the question of the evolution of intelligence. Is intelligence something that has only evolved once? Let's hear Gould's view on this.
Actually, it's really a curious phenomenon. If intelligence was meant to be, you would think it would evolve convergently in lots of other lineages.
Yes.
So is that right? So are we the only intelligent species? Has intelligence only evolved once? Has it not evolved in multiple convergent lineages? This is a phylogenetic tree I've constructed based, just for convenience, on mitochondrial proteins. Mitochondrial proteins not thought to have a particular role in the evolution of intelligence, so it's a fairly independent marker. Have humans at the top, tarsiers and mice we wouldn't necessarily consider to be on a par with us in terms of intelligence. Dolphins are not closely related to us. Well, we wouldn't necessarily think of sheep and hedgehogs as being geniuses. And similarly here we have the Caledonian crow, Corvus moneduloides. Kiwi birds and turkeys aren't normally considered to be exceptionally intelligent. The octopus, I'll show you later on, whether you can decide for yourself if you think they're intelligent, compared to a particular species of mussel and a limpet, which we wouldn't normally consider to be remarkably intelligent. All of these examples are organisms which clearly are very separated from us, from each other, very diverged from each other in evolutionary terms. Is this a case of animal intelligence evolving convergently among multiple lineages? Well, you can decide for yourself. I'm just going to show you about a 5-minute clip of key experiments testing this in the octopus, the Caledonian crow, and a dolphin.
The sign of true intelligence is when an animal solves a new kind of problem, something that no member of the species may have figured out before. For the octopus, removing a cork from a bottle to catch a crab is child's play. It's almost exactly like lifting a rock to find a delicious morsel underneath. It does it by instinct. But what about opening a screw-top jar? There are no screw-top jars in nature, nor any instinctual response to help in opening it.
Look, he's exploring the jar. He's touching it.
The little octopus's inquisitive arms are almost as sensitive and agile as human hands as they explore the novelty of the screw-top jar. Here it seems the octopus is not using instinct, but cognitive reasoning to solve this problem, thinking, if the crab got in there, there must be a way to get it out. Peppino thinks through the problem until he succeeds.
It's a complicated task. The crow is familiar with the individual pieces of the puzzle. He's done each of them separately for a food reward. But this is the first time he's seen them arranged like this. 8 separate stages that must be completed in a particular order if the puzzle is to be solved. If the bird succeeds, it'll be a world first. He takes time to have a look. And then starts with the short stick. He finds it too short to reach the food. He then sets off to get the first stone. Got it. But he doesn't seem to know what to do with it. And another. He appears to be stuck. But now something seems to click. He dispatches the first stone. Then returns to collect the last one. Got it. The final stage. Success. 8 individual stages complete. The crow performed all 8 tasks in the correct order.
For 1 month of the year, schools of mullet migrate up the coast. The dolphins herd the schools into the shoreline, exactly where the fishermen are. Then, at the right moment, the dolphins make a signal, usually a distinctive dive, and the fishermen throw their nets to catch the fish beneath.
It's absolutely clear that the dolphins are calling the shots here. These lads are doing absolutely nothing. They just stand there with their nets. And it's only when the dolphin makes a very clear signal that they deploy them. So the dolphins are telling the fishermen how to fish. That's novel.
Just a few miles down the coast, the same mullet schools migrate. But because no dolphins help the fishermen there, they hardly catch any fish. Here, the dolphins have developed a relationship over generations that's resulted in a thriving local industry.
The fishermen are benefiting. Why on earth are the dolphins doing this?
We expect that when the fisherman threw the nets in the water, he's also pushing the mullets to the dolphins. Much easier for dolphins to get the mullets.
When the net's descending, some mullet are inevitably going to be escaping, and that's the bounty that the dolphin can enjoy.
Exactly.
So it's a mutual, symbiotic relationship going on, dictated by the dolphin, right?
Yeah.
I mean, they call the shots.
Exactly.
So hopefully you'll be persuaded by those 3 examples of the common octopus, the New Caledonian crow, and bottlenose dolphins, that intelligence has evolved independently in multiple lineages. In these species, we can see examples of intelligence that is not simply explained in terms of instinctual behavior, but rather which reflects a degree of forward planning and strategic thinking. Now, I wouldn't want to misrepresent the views of Richard Dawkins. He clearly understands evolution by natural selection as a non-random process. But that doesn't necessarily mean that he understands it as reflecting any sort of higher purpose or design. Nevertheless, the independent evolution of intelligence in different lineages raises interesting questions as to whether or not the process of design is something unique to humans. Let's hear Richard Dawkins expressing his views on this subject first.
The human eye looks as though it has a purpose, but it has come about through the process of non-random natural selection. There is no purpose in the human sense. There's a kind of pseudo-purpose. Why do I use the word design only for human creations and not for the manufactures of these animals? The difference is that human designs get their goodness and efficiency from conscious human foresight. Wasp pots and ovenbird nests get their efficiency directly from natural selection by a kind of hindsight rather than foresight.
So according to Richard Dawkins, only human beings are capable of design, despite the evidence that we've presented that the common octopus, New Caledonian crows, and bottlenose dolphins are capable not merely of hindsight, but also of foresight in order to obtain food. That they have some degree of strategic design in their approach to acquiring food. In this respect, Richard Dawkins differs from the views expressed by Charles Darwin. Charles Darwin, writing in his book, The Descent of Man, described how the difference in mind between man and the higher animals, great as it is, certainly is one of degree and not of kind. For Charles Darwin, there couldn't be a clear categorical boundary between humans and other species, but rather there's a graded series in which we see traits such as intelligence. Now, this raises interesting questions. If intelligence and design evolved independently in different lineages, not just unique to humans, then could the process of evolution itself be capable of producing things that we might consider to be reminiscent of design? As you consider this, I'd like you to watch a clip from a presentation given by Professor Steve Jones of University College London.
I spent a couple of months in what was called the detergent shed. And to make detergent, to make washing powder, the principle is simple, the practice is much more difficult, but what you do is you take a huge vat, not quite as big as this hall, but big, and you fill it with a boiling hot chemical liquid under pressure, and you force that liquid through a nozzle. And as it comes out of the nozzle, it bursts into two phases. It bursts into a vapor, which is taken away by fans and recirculated, and a powder, which is your famous washing powder. And in my day, the nozzles looked like this. They were about that long, made of stainless steel, heavy, rather pretty, beautiful things, expensive to make. And they were useless. They kept getting blocked. They made grains of different sizes. One nozzle would only work with one particular formulation of chemicals. They were really very, very primitive things. So what the factory owners did, and did it across the world, was to hire a series of intelligent designers, really intelligent designers, mathematicians, physicists, giant brains of that kind, to try and improve this. And they had very limited success because the physics of moving from a liquid to a solid plus a powder, a phase transition as it's called, is very hard to understand. Almost without realizing it, I think, at the time, the engineers and the designers then moved on to classic Darwinian natural selection. What they did was to make 10 copies of that nozzle and mutate them, to change them slightly. At random, longer or shorter, bigger or smaller hole, different angle of attack as the liquid went in, and so on. And they tested them against each other. And maybe one of them was marginally better, 1% better than one had gone before. So they took that one, allowed it to reproduce, made 10 copies of itself, changed it at random, and carried on the process. It inherited differences in the chances of reproduction or natural selection, and very quickly they saw a remarkable change. Here's a rather primitive model of what happened. Over the generations, they began to evolve, and I use that word advisedly. They began to evolve through natural selection, a highly unlikely, almost impossible nozzle, which looked like that, and worked 100 times better than what had gone before. And nobody designed that. There was no foresight. It happened by the simple Darwinian machine. So it works. And it works on inherited differences. And the comparison is quite noticeable. That is what natural selection can do. It is a factory for making almost impossible things.
So if that is the case, is it completely unreasonable to potentially discern a purpose or a design in natural selection?
We'll come now to the elements to which chance might be involved. What we might describe as chance in the evolutionary process. Natural selection isn't the only mechanism for genetic change. One of them, another, is a genetic drift, which I'll illustrate briefly here. This applies to when you have very small populations only, where stochastic events, seemingly random events, can produce radical changes. So let's take this box here containing pink piggy banks and yellow rubber duckies. If you take away about half of them away diagonally, you'd be left with this. If they were to self-replicate somehow, I don't know exactly how piggy banks would reproduce, but if they self-replicated, you can see the population that would result. Again, we're going to just take a section of these away to another box, allow them to self-replicate. You can see how we're now getting a bias in the population. And then finally, take a corner out here, pick that population, and then suddenly we have a population existing only of ducks. This is genetic drift. And this tends to recur, as I say, when you've got small populations. You can have a genetic bottleneck, as occurred, for example, with the northern elephant seal, where their populations were reduced down to basically double figures alone to populations in California, and they have very little genetic diversity now as a consequence. Also can occur in something called the founder effect. So some of the early Dutch settlers, the Afrikaners in South Africa, they would tend to have an increased frequency of Huntington's disease because of presumably a higher prevalence of that in a smaller population that initially founded the Afrikaner population in South Africa. Mutations are often also considered to be rather random in terms of their occurrence. Here we see sort of various forms of irradiation flying in onto chromatin. I won't spend a lot of time on this, but just to give a few examples. Here we have an example of how methyl nitrous urea might potentially deaminate cytosine after it's been methylated, as will normally occur, and give rise to a different nucleotide-based thymine, or alkylation, ethylation by ethyl nitrous urea, or ethyl methyl sulfonate of guanine. So you can have various mutations occurring in response to chemical elements. Another example is ethidium bromide intercalating in between DNA in the major groove. UV irradiation producing thymidine dimers, which can produce a kink in the DNA, and this can also produce a kink, and a kink can sometimes result in propagation of repeats within the DNA, as can occur here in the propagation of a trinucleotide repeat expansion, such as occurs with this EAG repeat in Huntington's disease. So these are often described as random effects, usually because we don't necessarily know what the underlying cause is, but they are clearly uncaused events. So, question, what is truly random? Is this random, this sequence of 30 letters, or this one, or this one? Only one of these is actually random. In fact, it's the top one. This one is the first 30 amino acids of a particular protein I worked on, and this is a nursery rhyme recoded, replacing each letter. It basically just says Jack and Jill went up the hill, replacing every space with the letter A and every letter with the one that comes after it in the alphabet. It wouldn't necessarily be very easy to tell which of these is random looking at it. And so as this Dilbert cartoon illustrates, randomness, true randomness, is often rather difficult to recognize. So finally, what does the Bible say concerning all of this? Well, this is what it says in Proverbs chapter 16, verse 33. Behek yutal et hagaral ume Adonai kol mishpato, which translated as the NIV renders it, the lot is cast into the lap, but its every decision is from the Lord. So from a Christian theistic understanding, nothing is beyond God's realm somehow. God is behind it all. Now, there are right and wrong ways of interpreting this. If we're going to say that chance events, God is still behind them, one way of interpreting this might be, as in this slide that I borrowed from a young Earth creationist friend, where he would always be asking the question of where is God intervening? Some people might think, well, chance events are where God is acting, but that is really limiting God's activity. That amounts to God of the gaps.
The term God of the Gaps was originally coined by this person. This is Charles Coulson, who is the founding professor of theoretical chemistry at the University of Oxford. He was also a devout Christian. In fact, he was a Methodist lay preacher. And Charles Coulson said the following: "When we come to the scientifically unknown, our correct policy is not to rejoice because we have found God. It is to become better scientists." Even before Charles Coulson, this fallacy of God of the Gaps was also expressed by Dietrich Bonhoeffer. When he was imprisoned by the Nazis, shortly before he was due to be executed, he said the following in a letter to a friend: "How wrong it is to use God as a stopgap for the incompleteness of our knowledge." And he continued, "We are to find God in what we know, not in what we don't know." This fallacy of seeing scientific knowledge, knowledge of the world, as somehow competing with divine activity can also be explored by comparing different passages in the Bible that describe how a seed grows. So, for example, in the Gospel according to Mark, chapter 4, verses 27 to 28, we read the following: "Night and day, whether he sleeps or gets up, the seed sprouts and grows, though he doesn't know how. All by itself, the soil produces corn, first the stalk, then the ear, and then the full grain in the ear." So according to this passage, what makes a seed grow? The soil does it. However, if we look at Paul's first letter to the Corinthian church, in chapter 15, verse 38, we have this description: "But God gives it a body as he has determined, and to each kind of seed he gives its own body." So, according to this passage, how does a seed grow and develop? Well, God gives it a body as he has determined. Is it the soil then, or is it God determining a seed's development? Well, these aren't competing descriptions, they're complementary descriptions. The soil is necessary for a seed to grow, but ultimately it is God who determines its development. And we might understand this nowadays in terms of how genes influence development. In fact, when we go through the pages of the Bible, we have repeated descriptions of how God is not merely the creator of the universe, but how he ultimately lies behind what we might describe as laws of nature, because he is the sustainer of the universe. So, for example, in Nehemiah chapter 9, we have a prayer, and in verse 6 it begins like this: "You alone are the Lord. You created the heavens, the highest heavens, and all their starry host, the earth and all that is on it, the seas and all that is in them. You give life to everything, and the multitudes of heaven worship you." You give life to everything, you preserve everything, you sustain everything. God is the sustainer of everything in the universe. Similarly, when Paul is writing to the Colossian church, in chapter 1, verses 15 to 17, we read the following, as a description of Jesus Christ: "He is the image of the invisible God, the firstborn over all creation. For by him all things were created, things in heaven and on earth, visible and invisible, whether thrones or powers or rulers or authorities. All things were created by him and for him. He is before all things, and in him all things hold together." Here again, that description in verse 17. He is before all things, and in him all things hold together. God is the sustainer of everything in the universe, not merely the creator. And similarly, in Hebrews chapter 1, verse 3, we have another description of Jesus Christ as God. The Son is the radiance of God's glory and the exact representation of his being, sustaining all things by his powerful word. So, here we have a description again of God as sustaining everything in the universe. However, Jacques Monod had a rather different understanding. In chapter 2 of his book Chance and Necessity, Jacques Monod said the following: "All religions, nearly all philosophies, and even a part of science, testify to the unwearying, heroic efforts of humanity desperately denying its own contingency." Really? All religions? Nearly all philosophies? Where is Jacques Monod getting this from? Clearly, the picture we have in the Bible is of God as sustainer of everything there is in the universe. So our very existence is contingent upon God's providence. And this isn't a description that is unique to Christianity. As we've heard from passages in the Hebrew Scriptures, like Nehemiah chapter 9, verse 6, we have a description of God who sustains everything in the universe. In conversation with Muslim colleagues as well, I can appreciate that this is an understanding common to Islam too. So for all of the Abrahamic faiths, we have an understanding of how our existence, our very existence, everything in the universe, is contingent upon God's sustaining activity and his providence. Let us conclude, finally, with some words by Simon Conway Morris on this subject.
But if there is a God who's made the universe, then I think it's fair to say that if metaphorically he was to take his mind off it for a second, we would no longer exist. We are entirely supported in God's hands.
Thank you very, very much for that very thorough presentation, Neville. I think it's fair to say you've presented enough evidence and data that will be converging on your conclusions as well. So just as people start to make some more comments, a starter from me. So this is a quite a big area, and it obviously touches on a lot of areas of science, as well as philosophy and history and theology. So there's a huge scope of material to work with and to think about. So what is it about it that particularly appealed to you to kind of get stuck into it?
To get stuck into it, well, what prompted this presentation to begin with, I was asked to give a talk at the Christians in Science Student Conference back in 2017, where the general theme was to do with chance, and other people were addressing the question from a perspective of physics. I was addressing it from the perspective of biology. I suppose there's a number of things that appealed to me. One is just getting into the span of the Bible, seeing God's activity through that. Also, just the numerous examples of evolutionary convergence that we can see. I can see a certain level of beauty in that, particularly in the structures that are produced, which are more obvious perhaps in the various animals where we're looking at the whole body. But even at the molecular level as well, where we can see some level of pattern as well, I can see a degree of beauty in that too.
You gave some examples of the kind of the macro world, plants and animals, and you gave some examples of convergence at the genetic level and at the molecular level. Can you give a few examples maybe of somewhere in between? Kind of any examples of convergence in the kind of microorganism level, maybe kind of like bacteria or viruses?
Yeah, I mean, the long-term
carries on all the way through.
Sure. I think the long-term evolution experiment conducted in E. coli is probably the best example we have there, where experimentally we can see convergence in cell size, in patterns of gene expression, in positive selection for looking at the ratio of non-synonymous to synonymous substitutions. There's clear positive selection occurring and convergent evolution of different mutations occurring in different proteins. So probably that provides the clearest example.
So we've got a question from one of our watchers. Stephen J. Gould refers to not just chance, but also good fortune. Why good?
Well, unfortunately, Stephen Jay Gould is no longer alive and with us. Otherwise, he would probably be the best placed person to address that question. I mean, Stephen Jay Gould himself was a person who had a clean sense of ethics and morals. I mean, he's coming from as a non-believer, nevertheless culturally from a Jewish background. He had very strong views regarding particularly attempts to categorize intelligence as something heritable with political implications. So he wrote strongly against Murray and Herrnstein's book, The Bell Curve. He was somebody who had a strong moral instinct. I suppose if he was pushed to it, where those morals ultimately get their authority from might be a more challenging question. But really, that's a question that only Gould himself could address. And sadly, he's no longer alive and with us.
So a question from me that you will probably not be surprised to hear. Would you expect a same kind of direction of convergence in a totally novel system, such as maybe an extraterrestrial ecosystem? So something that has not been derived from our ecosystem at all. Would you expect the same pattern of convergence to emerge there?
It seems probable. I mean, if, say, another ecosystem has similar laws of physics as we would expect would hold true throughout most of the universe, then I would expect to see similar patterns. So I'd see physical constraints as being largely responsible for a lot of the patterns of convergence that we see. And so insofar as the laws of physics are conserved across the universe, then I would expect to see that. It's interesting, at the end of his book, Life's Solution, Simon Conway Morris suggests that if aliens were to arrive in the spacecraft, they probably would have camera eyes like us. Now, of course, that's wild speculation, but it indicates the strength to which he thinks that they will resemble us and not necessarily be bug-eyed, as he puts it.
Yeah. So how would that integrate into a kind of a Christian theistic kind of worldview in terms of kind of like, if we've got multiple cases of convergence, is that consistent or inconsistent with a God who creates here on Earth?
I don't think it really has strong implications either way. So, for example, C.S. Lewis writes a short novel, a sort of a short chapter in one of his books, God in the Dock, it's kept in, where he sort of suggests that people almost want to try and have it both ways, and suggest that if we are the only life existing in the universe, and that's somehow significant, and yet if there's life on another planet as well, then that's equally significant. And you know, I think it's a bit of a non-starter in many respects. I mean, the life here is remarkable that we have on this planet. It's the only planet that we're aware of strong evidence for life. That's not to rule out the possibility that we might discover some exoplanet in the future with some evidence of, at the very least, microbial activity suggested by the nature of its atmosphere. But you know, it's pure speculation at the moment. We can't say strongly.
So another question from a viewer. At what level is chance operating, or does God control absolutely everything, e.g. if an individual might fall off a cliff, for instance?
Well, there we come down to the question of free will and determinism, a deep philosophical question, which people have still been debating. And some people would see the two things as not necessarily competing with each other. So, for example, Daniel Dennett describes himself as a compatibilist. He's a philosopher, an atheist himself. And he would argue that free will is somehow compatible with determinism. Now, of course, working out the details of that is another matter entirely. But all of us would have an instinctive sense of right and wrong. If you were to injure somebody in some way, they wouldn't be likely to say, oh, that's just down to chance, it's not your fault, or anything like that. We usually have some sort of sense of responsibility, which we would root in an understanding of free will. We tend to live our lives that way. Is it down to chance, or is it down to God? Is it an either or? I think that's one of the difficulties. If we're to take, for example, from Proverbs 16:33, which I quoted during the presentation, the lot is cast into the lap, but its every decision is from the Lord. It's seeing that even the chance events, somehow God is ultimately behind them, ultimately responsible. And if we understand God as sustainer of the universe, then that makes sense. It's not to say that God is only behind chance events, but neither is it to say that chance events are the alternative to God's activity. It does raise, though, interesting unresolved questions regarding where free will fits into that. And philosophers have debated that for ages, and I'm sure they will continue to do so.
It keeps them in a job.
It does.
Another question. Do you see the idea of underlying patterns directing convergence in evolution as an indication of design and purpose, or is it simply utilitarian, what works in the context of this Earth?
I think it's both. I don't think it's an either or, to be honest. In terms of survival of the fittest, yes, it is utilitarian, but why should such a universe exist to begin with in which natural selection can operate? there is some degree of purpose in natural selection, or at least there is some degree of pattern. And if we understand, and clearly I'm coming from a very specifically Christian theistic perspective here, if based on all those passages in the Bible that I illustrated, we understand God is ultimately responsible, ultimately sustaining everything that there is in the universe as well as creating the universe. If we understand God as sustainer, then it's not an either or.
So I got a question, which is less about the actual science of convergence, and more about the kind of educational side of it. So why do you think examples of convergence, such as what you've outlined tonight, haven't entered the public and the church imagination around discussions of evolution?
To some extent they have. I mean, if I was to look through older books of natural history that I sort of grew up with, and some of them were produced long before I was born as well, but I inherited them from family members, you would see examples of convergent evolution, particularly among mammals, particularly highlighting how various marsupials and placentals converge to have similar patterns. So I gave the example there of the marsupial mole, the thylacine convergently evolving with burrowing animals and wolves. Many other examples could be listed as well, hopping animals, etc. So they have been included in some books that have been distributed for the general public. As to why it hasn't entered the public imagination more, it's a good question. I don't really know. It's not that it's been completely ignored. And certainly Simon Conway-Morris has made strong efforts to list all of the examples in his book, Life Solution, and some of the research work that went around that. He's collated together numerous examples. I've tended to focus more on some of the ones at the molecular level, whereas Simon Conway-Morris will focus more at the level of the whole organism because of his background as a paleontologist. So there are plenty of examples.
What about in the church itself? Do you think it's a kind of, because it strikes me as a very powerful, not necessarily hardcore proof, but a very powerful pointer to kind of some kind of design and purpose behind what we see around us, and that has a lot of resonance for kind of Christian theologies of creation. Do you think it's just a kind of general tepidness towards evolution in some parts of the church that's presented?
That could be part of it. Yeah, that could be part of it. But of course, we don't want to sort of jump to conclusions too quickly either. I mean, what convergence illustrates is that the process of evolution is not entirely random, that it is constrained. So there are people like Richard Dawkins, for example, who would fully agree with that, but wouldn't take an extra step of seeing it as indicating design and purpose. Yeah. But certainly in terms of a more nihilistic perspective that sees evolution as without any pattern whatsoever, convergence provides numerous counterexamples.
It could be an interesting point to try and promote a bit more that it is non-random, because often a charge against evolution in the church is that it is a random process, and therefore God is not present in the process.
Indeed. But also it raises wider questions as to what actually is random. And as illustrated there with a few trivial examples in the Dilbert cartoon as well, it's not always very easy to recognize true randomness.
Yeah.
So I think we have to have a certain degree of caution there.
Yeah.
Even like with some computer programs that I used to run for looking at molecular phylogenies, where you're using maximum likelihood methods, you'd have a random number generator to give you a seed for the program. And there was a particular random number generator I would use where they were looking at various atmospheric variables. But your computer doesn't really give you a truly random number. If you're to enter in an Excel, sort of get rand X, it's not truly random.
Yeah, we had a presentation in one of our talks on this on the Manchester group last year from Rhoda Hawkins at Sheffield University, where she talked about randomness being behind and necessary to certain physical outcomes. Little plug there, you'll find it on our YouTube channel. So a couple more final questions that I usually ask our speakers. So how has your thinking in this area impacted your own personal faith?
I would say it's probably in terms of recognizing God's sovereignty. Recognizing him as a sustainer also has a recognition of his sovereignty and his purpose, even if I don't necessarily see immediately what that is. So in the same way that the first 30 letters that I presented, which actually the first 30 amino acids of the SMC4 gene from Drosophila melanogaster, you might necessarily recognize any clear pattern in that to begin with, but there is a pattern there. And similarly, even if I don't always fully understand what God is doing in any particular instant, I can still have a trust that he is sovereign and has some purpose.
And where do you want to see science faith engagement develop? Just a general question.
Yes, I think probably more conversation. Conversation is good.
Excellent. Fully appreciate and approve of that. And just the final question, what books or resources would you recommend on your topic for people wanting to dig in a bit further?
Well, probably Simon Morris's book, Life Solution. That was published quite a while ago now, but I think it's still a lot of the comment, and that's still is still relevant.
Mm-hmm. Anything on the theological side, particularly?
On the theological side, I'd strongly recommend any book by John Polkinghorne. Sadly, he died last year, but he certainly left books that are fairly accessible, fairly slim volumes. That would be one that I would definitely recommend.
Excellent. There are some comments saying thank you very much for your presentation. People seem to have really enjoyed it and appreciated the depth in which you went to in it as well. So I think we could all applaud at our computers, and I'm sure Neville would appreciate the thought as well. So thank you very much, Neville, for your presentation tonight.
Thanks, Gavin.
So we are clocking on, coming up to 10 to 9, or just gone 10 to 9 now. So it's probably time for us to wrap up. Thank you all for joining us as well tonight. I hope you found the new format helpful. I think it's worked quite well at my end of things as well. So just to remind you that our next event is on February the 25th. So that's just over 2 weeks' time at 7:30, where we'll be joined by our development officer, Steph Bevan. So put that in your diary now, February the 25th, and then also March the 31st for Joshua Smith coming over to us live from Mississippi on robot theology. You can also go to our Eventbrite page to register for these events. They'll both be going up in the next couple of days. So keep in touch with us and keep informed on what we're doing there. With COVID cases still remaining high at the moment, just for now, we're going to keep meeting online. But there is discussion in Christians in Science about a kind of linked up meeting where we're going to try and link up all the local groups live for a presentation coming from the Faraday Institute down in Cambridge. And we're looking to do this maybe in May if things work out for that. Also on May the 7th is a provisional date for the next CIS autumn spring conference, what we used to call the Northern Conference, but it's now going to be called the Spring Conference. This is looking like it will be at St. John's College in Durham. And the theme for that conference will be public engagement. That will probably be a hybrid event where we'll be broadcasting online, but we'll also be meeting in person. So thank you again to Neville for his excellent presentation and his time preparing that. I'm certainly going to have to go away and digest it some more. So until we see you again, take care and good night.